welcome to the class today. I'm sorry about the delay. Today we'll be talking about a different method, which is expansion microscopy. How many of you have heard about this method? No? Oh, somebody did. Okay, so we're not only going to be talking about expansion microscopy, but we'll talk about the newest development of this methodology. And the two references that I listed here, one of them is from Nature Methods. This is published earlier this year, but this is only a one-page summary or preview of this paper on Nature Biotechnology, also published earlier this year. So the second reference here is the main methods paper that we will discuss today, but we will start by talking about expansion microscopy altogether. And actually, we'll start by defining the terms. So resolution is a word that has a lot of different potential meanings, right? So it could be an action to solve a problem. It could be an agreement that's achieved by multiple parties of diplomats, right? It could be, we're approaching the end of the year, so you can be asking your classmates, what's your New Year's resolution? That means what do you want to achieve in the next year, right? But in microscopy, resolution specifically has a specific definition, which is the minimal distance between two distinct points that can be seen as two separate entities by the observer or the camera. So in the example here, we're trying to view the objects that's in orange. And if the distance between the two orange objects is larger than the resolution of this specific microscope, then under the scope, you will be seeing two separate foci, right? Because each of these places will emit fluorescence that's illustrated by these blue lines, and then you will see 1 and 2. Okay, so no problem with that. However, if you're trying to view two objects that's very close to each other, so if their distance is smaller than the resolution of this microscope, then even though they still emit separate lights, it's illustrated by the dashed lines here, but what we will view and what we will see is only one combined intensity. So you will, instead of seeing two separate objects, you will see one foci or one bright point in the microscope. It could be slightly broader, but you won't be able to say, okay, this is two different objects, right? So for this reason, scientists has been trying really hard, trying to push for smaller and smaller resolution in microscopy, because a lot of things that we're interested in, we want to be able to distinguish the objects that are separated within a very small distance. All right, but in optical microscope or optical microscopy, the resolution is not, you cannot push the resolution beyond what we call the diffraction limit. So this is not a physics class, so I won't go into how this was determined, but the diffraction limit is denoted by D here, and it's equal to the wavelength of the light that you're using divided to 2 times the numerical aperture of the lens that you're using. Okay, and then here I'm providing the rough ballpark of what we usually have in optical microscope. So if you're using, blue light illumination, your wavelength is about 400 nanometer. If it's red, far red, then it's approaching 700. And then the numerical aperture of the lens, if you're using a really good lens, then you have an NA of about 1.4 or slightly above that. But if you do the math here, you'll get a number that's about around 150 to 200 or even 250 nanometer, which is the diffraction limit. Okay, in normal optical microscopy, we cannot achieve a resolution that's lower than the diffraction limit. And that could potentially limit what we can do in the microscope, right? Because, of course, a lot of us do cell biology and we're interested in the cell. The cell is bigger, but the diffraction limit actually falls around the size of an organelle or structures that's within the organelle, which a lot of times is what we're really interested in looking at, right? But when your resolution cannot be lower than this number, then it could be a problem. And of course, for even smaller stuff like, protein assemblies, atoms, you need to do, other kind of microscopy like electron microscopy and something else. All right, but for biological scientists, a lot of things that we're interested in falls within that range that's slightly beyond the diffraction limit, right? So the scientists have been trying very hard to break the diffraction limit. And the first series of technologies that has been developed since the 80s till now is super resolution microscopy, which you may have heard of. And then there are different kinds of super resolution microscopy. It's a very mature technique. There are different variations and a lot of different variations, which I won't go through. But long story short, many super resolution microscopy uses different illumination patterns or different ways to activate or deplete fluorophores to achieve a resolution that's smaller than diffraction limit, right? And then it's so many technology that's in this review paper that's published a few years ago in Nature Cell Biology. They provided a selection tree for super resolution microscopy. So if you have your biological question or a specific structure you want to view, just follow this tree, and then it will give you recommendations of, okay, what kind of super resolution microscopy is going to help you with your experiments, right? Do you need live cell imaging? Do you have a lot of money to buy a new scope? Do you want to stay with what's commercially available already? Or, what kind of algorithms you can use, other things that's available. Yeah, so there are a lot of different options for you. But the problem with super resolution microscopy is that you need to buy a very expensive equipment for the most part, right? And then imagine that even if you have the best super resolution microscopy, in a few years, if a next generation has been invented, then do you have the money to buy the next newest, the best thing, right? So that will be a problem for smaller institutes or labs that doesn't have a lot of funding, such as my lab, right? So for similar reasons, there are another approach that's been developed by groups of scientists, which is, okay, when we try to break the diffraction limit using super res, we're trying to make the microscope better, right? But how about if we, instead of trying to make the microscope better and spend more money in the microscope, how about if we just make our sample bigger, right? So even though you don't actually make the resolution better, but if your sample is bigger, then you can use the same old microscope that you already have in the lab, but use it to view things that used to be very tiny, right? So if you make it bigger, then you can view those things. Those things are now going to be bigger than the diffraction limit, and then you can see them with a really ordinary microscope. So this concept is new and elegant, and it's a simple idea. And then the next question is, how do you achieve this, right? And of course, if you're lucky and you happen to have a robot cat that was sent back to help you from the 22nd century, you can just ask Doraemon, be like, Doraemon, help me with this. I need to magnify my samples. And then Doraemon's going to pull out this magnifying lamp, and then problem solved, you're good, you graduate in 3 years with your PhD. Okay, unfortunately, that's not the case, right? So scientists have to figure out how they can expand their samples and still be able to view them under the microscope. So what they come up with is the polymers or the hydrogels. Basically, the idea is that, okay, they're going to anchor the biomolecules they're interested in into this network or meshwork of hydrogels. When the anchoring happens, the hydrogel is in a shrunk stage. So the hydrogel has the potential to expand, but it was in a shrunk stage when you first associate your samples with this hydrogel. After cross-linking and everything has been done, you will do a magic trick to expand the hydrogel. So now all these like shrunk parts of the hydrogel would expand and then becomes something like that. Naturally, you can expand your sample. Okay, and then this is an example here. You're going to see a lot of mouse brain today. This is a piece of the mouse brain, and this is before expansion. Panel C is after expansion. So you will see that they become a lot bigger with the same scale bar here. Also, you'll notice that it goes from a solid piece of slice becomes something that's transparent. Okay, and we're going to come back to that point because that actually makes a huge difference in the methodology. Okay, so the concept is simple, and after a lot of trial and development, this is what was reported a few years ago also on Nature Methods. This is how people have been doing expansion microscopy. So first, they need to anchor the biomolecules of interest to an anchoring agent, which is this pink thing here. So for example, if you're interested in visualizing a protein or different proteins in your sample, you have to anchor all the protein to this molecule. And this pink anchoring agent is a molecule that's able to interact both with the biomolecule that you're trying to see and the hydrogel that you will synthesize later. Okay, so you apply this anchoring agent, and this is usually a very time-consuming step, so it will take many hours or even days for this to be complete. But you need this time to allow for a more complete anchoring. Otherwise, even if you can expand the sample later, you won't see what you want to see, right? So anchoring happens. And then you add the polymer or the monomers, which can be synthesized into polymers in your specimen, and allow the polymer to form. And then in step 3, you need to do something called a mechanical homogenization. This is the step that breaks the existing bonds between your specimen. Because you can imagine if the cell is intact and you try to expand the cell, you're going to break the cell, and the molecules that you want to view won't stay in the same relative position once you expand them, right? So you need to basically break those bonds. This is achieved usually by enzymatic digestion, so you can use proteinase K or some other proteases to break all the bonds, including some of those mechanical like resistance in the extracellular matrix. Okay, so, and this is also a step that you can try, you can use different protease, different procedure, protocol to try to optimize it. After homogenization is done, you go to the final expansion. And because it's a hydrogel, so the expansion is basically soak your specimen into water, right? And then the polymer will expand. And then that completes the expansion microscopy process. Now you'll notice that even though the name says expansion microscopy, it actually doesn't involve, inventing a new scope, right? You can use almost any scope to view your now expanded samples. Summarizing the outcome of the sample expansion, this was first reported in 2015, and up to a few years ago, expansion microscopy can expand your sample to about 4.5-fold in one dimension, which means that if you time this to a 3D, 3 dimensions, you get about 100-fold expansion in terms of volume, and that's a lot. Also, because of the homogenization process, the sample becomes clear because now it's 99% water, and that provides some advantage, which we'll come back to in a few slides. All right, next I'm going to show you some examples with expansion microscopy. This is taken directly from the first paper that was using this method that's published in Science in 2015. So on the left here, we're viewing some microtubules. And this is already a super res microscopy. So you will see that you can see the microtubules quite well. But not as good as this middle panel where you are using a normal or regular confocal to image an expanded sample of the same microtubules. Right? You'll notice that a lot more details are provided, especially if you focus in the zoom in area and like do a line scan along this dashed line. So in the super res image, you will see only one peak, even though by visualization, you'll be like, okay, I think there are 3 different microtubules here, right? But if you do a line scan, it's not really clear. But in the expansion microscopy, you are going to see 3 different peaks, meaning that this method allows you to differentiate these 3 structures. Okay, the second example I'm showing is when a scientist wanted to visualize the clathrin-coated pits in the process of endocytosis. Again, they're comparing the super resolution versus the confocal image of expanded sample. And you'll also notice that a lot more detail is provided in the expanded sample. The panel on the right is kind of important because it's comparing the radius of each individual CCPs before and after expansion. And if the expansion is truly isotropic, meaning that in every dimension it was expanded equally, then you expect every single one of them to fall on this diagonal line, right? So you will notice that it's not perfect, but it's very close. You do see some expansion that's not isotropic, but the difference is small, and scientists on average think that the difference is about 2 to 3% after expansion. All right, and here's the mouse brain again, and I had to rotate the image so that it will fit the slide, but on the bottom is the original slide. And then the expanded sample is on the right. You'll notice that if you use a very small magnification, then it doesn't look that much different. But as you zoom in more and more, you'll notice that the expanded one provides much better resolution. And here, the boxed region, hopefully you can see, is standing of 2 different proteins. One of them is presynaptic. The other one is postsynaptic. So hopefully, in the ideal world, you'll be able to see them like side by side next to each other. But you'll find that it's a detail that can only be provided in the expanded sample and not in the original sample. All right, so expansion microscopy provided a lot of detail, and people have been developing newer and newer methods based on this. In the 2019, it was summarized that not only you can do expansion microscopy, but you can do iterative expansion microscopy, meaning that you can expand the cell or the specimen for more than once. So in this particular experiment, they basically performed the protocol and expand the specimen, but they found a second hydrogel or polymer that they can use. So then after the first round, they just repeat everything again and expand the specimen again. So if you do this twice, you can end up, expanding your samples a lot, and the resolution that you will get is going to be about 25 nanometer, which is about the best you can get in super resolution microscopy, or even better than that, right? So different super resolution microscopy is going to have different resolution, but 25 is, about the best you can get in any microscope. And here is the same 2 protein that we were viewing as the last slide. So one of the protein is called Homer1, the other is Bassoon. And not only in this picture, not only they can see that they are lining side by side, but they can also do a third channel and then see glutamate receptor on one of the membranes. And again, if you do a line scan, the peaks are really well resolved. Okay, and then we've been talking about the clearing of the sample by expansion microscopy, right? Why is the clearing helpful? Well, for one reason is that for any microscope, if your sample is transparent, it makes your life a lot easier, right? That's why people do a lot of microscopy on zebrafish, right? Because they're transparent. Another reason that the transparency helps with the microscopy is that people can now do something called hybridization chain reaction. This is a method to amplify the signal, especially when if you're doing fish, so fluorescence in situ hybridization. If you're familiar with fish, you know that you would add the probes to the cell, and then it will interact with the mRNA that you want to visualize, right? If you can do hybridization chain reaction, that means that on your fish probe, in addition to the part that binds to the mRNA, you have this purple thing called HCR initiator. And then after the probe binds to the mRNA, you add another thing called the HCR hairpins. And these are hairpins that has some fluorescence, and it can be conjugated next to the HCR initiator, and then it can go on and on and on and on. So each mRNA molecule now can have multiple HCR hairpins that has signal. So instead of seeing a very dim signal in the original sample, you can now amplify the signal to be a lot brighter. But hybridization chain reaction needs space. Okay, so this is something that works a lot better in the expanded sample with your transparent expanded sample, right? So because the expansion allows you to have the space for all these reactions to happen. And the example here is, I think the top one is pre-expansion, and then the lower one is post-expansion. And then they are pretty focused on these green dots, and you'll see that pre-expansion, you do see green dots, but you only see the most bright ones. But post-expansion plus the hybridization chain reaction, you will be able to see a lot of the dimmer dots that wasn't viewable in the pre-expanded samples. All right, so far everything that I've told you is probably invented a few years ago. But there are some limitations. It's already pretty awesome, but there are some limitations about expansion microscopy. First, like I mentioned before, the anchoring step is really time-consuming. In addition, it's molecule-dependent, which means that if you're interested in looking at the proteins, you do a protein anchoring agent. If you want to see the mRNAs, you do an mRNA-specific anchoring agent. Those are not compatible with each other. It kind of limits how you use this. The second limitation is that for most protocols, it's having problem to expand some clinical tissues that's usually very rigid because they're fixed and embedded, right? So the biopsy samples are especially difficult to be expanded. And then the third limitation is that because you're expanding the sample, right? So fluorescent signal that used to be very close to each other now are very spreaded. And that kind of can affect how you can detect the sample or detect the signal or not. And the last limitation is that because of the nature of expansion microscopy involves, anchoring and homogenization, it kind of only works with the fixed sample. So there's no possibility for live cell imaging for expansion microscopy. Yeah, it would be actually very scary if we can do live cell expansion microscopy one day. Okay, so some of these limitations are not easily addressable, like the third one and the fourth one. The third one we can overcome by some methods to amplify the signal after expansion. The fourth one, really, there's no way to overcome that. But the paper that's published earlier this year addressed the first two limitations of expansion microscopy quite nicely. So what they did is to basically optimize the method of expansion microscopy, and then they come up with a new strategy called Magnify. So that's the name of the protocol or the methodology and the whole process that they decided to use. I'm summarizing what Magnify can do here. So it's an improved strategy for the expansion microscopy. Compared to the original expansion microscopy, Magnify actually combined the anchoring step with the gelling step, so then it takes a little bit less time, and the effect is even better than what people can achieve previously. In addition, Magnify also gives you a higher expansion factor. So it used to be 4.5-fold. Now with Magnify, you can achieve 11-fold. Protein retention means how many proteins can be anchored successfully and expanded, right? So Magnify also gives you higher protein retention. In some difficult samples, it could actually give you more than 300% of what was retained previously. Moreover, Magnify allows us to visualize the lipid, which was not achievable with the original expansion microscopy. And Magnify is also compatible with the biopsy samples that's rigid, fixed, and embedded, which was not usually very well done in the original expansion microscopy. So how do they achieve this better or optimized methodology? They spend a lot of time trying to find a better anchoring agent, and they end up finding this simple molecule here called mesacrolin. Mesacrolin used to be used as a fixing agent in some of the immunofluorescence long time ago, like in the 80s or 70s. But the authors of this paper found that mesacrolin is very efficient in terms of anchoring the biomolecules to the hydrogel. And when the anchoring happens, one side of mesacrolin is interacting with the biomolecule and the other side is participating in the hydrogel. And during this process, they also optimize the composition of the hydrogel that they're using. It used to be composed of, some SA, AA, DMAA, I don't even know what this is, and they have different compositions. But in addition to adjusting the composition, the authors of this paper also add bis, which is another cross-linker. And by adding bis, they found that it increases the efficiency of cross-linking / anchoring. So they modified the protocol. Now we see optimized method and anchoring agent. What used to be 2 separate steps now is only 1 step. So anchoring and gelling of the polymer is now 1 step. And so that speeds up the whole process. Another thing that's amazing with this optimized method is that now the anchoring agent can anchor proteins, nucleic acids, and lipids. So you don't need to think about what you're going to view afterwards. You just use the same anchoring agent for anything. That kind of simplifies the whole protocol and made it more broadly available for people with different purposes in their research. All right, so this is, again, the mouse brain slice. And what they do is that, yes, they look at the same group of proteins, and then in all kinds of different brain regions, they can see different, the Homer and bassoon being next to each other. They can actually measure the distance between these two, and then come up with the brief conclusion that the regions in the cortical and versus the regions near the center has different distance between the pre and post synaptic cells. And this is the biopsy samples that was very difficult to expand. And this is like patient sample from colon and breast cancers. Here they're viewing 3 different proteins, and then you'll also notice that pre versus post expansion. The post expansion always gives you more clarity, and it doesn't change the position of these different proteins too much, which means the expansion is still largely isotropic. All right, and this is something that really excites me because they can look at the membranes. The membrane used to be something that is only, you can almost only visualize that in electron microscopy, right? So if you can do membrane visualization in light microscopy with such clarity, that's a big helpful tool. So here they are using 2 different staining. The one is the total protein staining, is NHS, and the bottom one is the lipid dye. So using expansion microscopy with a higher expansion factor, they can see clearly the stacks of the Golgi membrane. That's very beautiful here. And if they look at ER or nuclear envelope, they can actually see the double lipid bilayer. Here, that's the nuclear envelope, and then that's the bilayer at the ER, which means that you can use a normal microscope to achieve this high-resolution image. All right, the last example that I'm showing here is when people combine magnified expansion microscopy and super-resolution microscopy. And what they wanted to do is they wanted to look at samples that was difficult to be analyzed by optical microscopy before. So they are looking at the cilia. This is primary cilia of different cells. This used to be something that people can only use electron microscopy to study, right? And the cilia usually, the normal cilia would have 9 microtubule doublets, and they are aligned well in a circular shape. But in some disease context, then you will see that sometimes it's not 9 doublets, it's 8 or 7, and then the morphology or the shape of the whole cilia and how the microtubules line up is also different. But this used to be something that people can only use electron microscopy to see. But with the magnified plus super resolution microscopy, you can see that the image is still, of course, not as good as the electron microscopy, but you can kind of count how many microtubule doublets are there, and you can see how the shape is different in those abnormal ones. So for some purpose, this is already enough, right? And that would increase the speed of study or researches that's relevant to celiogenesis or diseases relevant to defects in this process. So that's very helpful in terms of how we can do the research in an easier way. All right, at the end of this lecture, I will leave with a few questions that I think we can think about, because hopefully you're like me, like after learning about this technology, you'll be like, maybe my lab can use it, right? Maybe in your project you can think about using expansion microscopy. To me, the future of expansion microscopy could have more broader applications for sure, but there could be also potential problems that arise, right? Because just like when people are trying to get smaller resolution of the microscope, we will face different problems when we expand the samples, right? Because we cannot do it endlessly either. Most of the lens that we work with has a short working distance, meaning that it's best when you're viewing the thing that's very close to the lens, right? Imagine if you have a lens that has a very short working distance, but your sample is huge. That means even though theoretically you can see the things that's in the sample, but you cannot focus it well, right? So that's going to be a problem if eventually. So maybe for now it's okay, but if you're viewing something that's huge or something that's going to be huge after expansion, then this is something that you need to consider. Another thing that you need to consider is the distortion, right? So in principle, the expansion is isotropic, but it's also known that the distortion is proportional to how many you're expanding the sample. So the more you're expanding, the more distortion you're going to get. Like to what degree you're going to say, okay, we're getting too much distortion and this is no longer representing what happens in the actual sample, and when to say, no, this is too much, we should stop, right? So this is something that we will need to think about before we actually use expansion microscopy in our research projects. But overall, I still think this is a cool method that sounds like not too difficult to use, and hopefully some of us can get our hands on this method pretty soon.